Alright, hello everyone. Today we're going to be talking about light microscopy. Also, the Bedwars in the background is from before the update. I do not play Bedwars anymore. I'm literally boycotting that game. But anyways, today is not really a biochemistry video because I am out of that course now. I'm taking bio, so I'm taking 3 bio classes. And in one of my bio classes, we're talking about microscopy. And our first lecture was all about different types of microscopy. So I'll be sharing a brief overview of each kind of microscope. I thought it would help both my learning and for you guys to understand in case you guys ever have to learn about microscopes and have to do labs on them or anything. So yeah, let's get right into it. Before we start talking about the different types of light microscopes, I just want to make a little note. When we deal with microscopes, there's magnification and resolution, and magnification is basically how big or how small you're making it, obviously if you're zooming into a photo, but this does not equal resolution. Resolution is how high quality the image is. So when you think about like 2K quality versus 4K quality versus like 480p, that's different types of resolution and different types of pixels. So let's say there's like 1,000 pixels per a certain area versus 100 pixels per a certain area. Obviously the 1,000 pixels will have a more detailed picture because it has more color, it has more individual pixels per that same area, so therefore that would have the higher resolution. So don't get those two mixed up, the magnification versus the resolution. But yeah, anyways, we're gonna start talking about different microscopy. First one is just your standard light microscopy. As the name states, you use light, and it's usually your typical one you'll see in labs, and the ones at school especially, they're not the best unless you're using the oil drop, but it does give you an introduction to microscopy. So you have the eyepiece on the top, and then the objective lens, and then the ocular lens, and the condenser, and then under that is a light source, so it just kind of shoots up. And I'm sure you guys all know about light microscopy. It's the most common type you'll probably see mostly in like undergrad and stuff as well. But yeah, that one's just really general. I'm not gonna go into that one too much because we have a lot of other ones to cover. The second kind of microscopy is interference microscopy, and this one you can clearly distinguish because it has a high contrast or higher contrast than the other light microscopies. So there's 3 types of interference microscopy. There's the light microscopy, the one that we were just talking about, DIC, which stands for differential interference contrast microscopy, and then we have phase contrast. So we already know what light microscopy is. Light microscopy appears more 2D. It's your just standard general one you'll find in the lab or in your undergrad is most common one you'll encounter. But phase contrast has a more grainy look to it when you look through a microscope, and the image generates based on the degree of darkness or brightness, depends on the refractive index of that region, versus the differential interference contrast microscopy, which the contrast of it is generated by differences in the index of refraction of the object and its surrounding medium. So when we compare phase contrast versus DIC, when we see pictures of them, phase contrast obviously looks a little bit more grainy, whereas DIC looks more leaning toward light microscopy, but maybe like a slightly more 3D version of light microscopy. It's very hard to distinguish sometimes between light microscopy and DIC, especially if the image is in black and white, which it usually is on an exam. So my teacher told us that we wouldn't have to identify a light microscopy versus a DIC. She's not going to put them beside each other. So I assume that's not a very key concept to know what they look like. The only thing you just need to know is phase contrast is grainy, light microscopy and DIC are more 2D flat, where DIC you can see kind of more edges. So another type of light microscopy is fluorescence light microscopy, and as the name states, you use fluor. So fluor is this fluorescent molecule, and it's added to the cell by fixation or genetic engineering. It basically makes the cell glow, hence fluorescence. It's a very powerful technique. It really helps with the visualization of seeing subcellular structures and individual proteins. So basically we have this excitation wavelength that goes like kind of sideways usually, and then we have the specimen on the bottom that contains the fluor, which will glow, and then we have the emission wavelength that is visible. It goes kind of up in this diagram, I guess. And there's a few possibilities to get fluorescent molecules in the cell. So we can start with immunofluorescence. This is another subcategory of fluorescence microscopy, which is a subcategory of light microscopy. I know it's many different subcategories because there's so many different types. They use similar techniques but slightly different at the same time. So immunofluorescence, it's basically taking antibodies and making them glow. Like that's just the simplified version of it. So immunofluorescence uses fluorescently labeled antibodies. Antibodies are obviously immunoproteins. I think I touched a little bit about it in one of my previous videos about antibodies. If you want to go watch that, it's definitely applicable to this. And so basically antibodies are made to detect a specific antigen and binds to a specific molecule of interest. So we have primary antibodies and secondary antibodies in immunofluorescence. So usually primary antibodies are from like dead animals. They usually use mice or bunnies to produce primary antibodies. So basically you take a dead mouse or a dead bunny or whatever your specimen is, and you take the antibodies out of it, and that's called a primary antibody because it's from the source. And this antibody is able to bind to a target molecule And then a secondary antibody is basically an antibody that's specific to the primary antibody. It's able to go on to and attach to the primary antibody, and it creates this covalent link to the fluor. So basically think the secondary antibody can covalently link to the fluor to get that glow, and then the secondary antibody with the attached fluor on it is able to attach to the primary antibody, which the primary antibody has that antigen binding region. So then after we establish the primary and secondary antibodies, there's two possibilities for immunofluorescence. We can either do direct immunofluorescence or indirect immunofluorescence. Now in direct immunofluorescence, the signal is weak and this primary antibody has the fluor. So as opposed to what we were just talking about, which was kind of more indirect immunofluorescence with the secondary antibody, for direct immunofluorescence, we do not need a secondary antibody. Basically, just think the primary antibody from that dead specimen is carrying the fluor, which goes on to the molecule of interest. So it's basically able to directly attach. But usually with this, the problem is that the signal is very weak because we don't have that secondary antibody to covalently link. Whereas in indirect immunofluorescence, so that was direct immunofluorescence, indirect immunofluorescence has a very specific and very good resolution. It's basically what we were talking about with the primary and the secondary antibodies. You take the primary antibodies, so you have the primary antibodies and it binds to the molecule of interest with no fluor, and then a secondary antibody is able to bind to the primary antibody to create that fluor because the secondary antibody again is carrying the fluor. So secondary antibody plus the fluor goes on to the primary antibody, and the primary antibody is already on the molecule of interest. So it creates a bigger signal, like a very high resolution, and multiple secondaries are able to bind to the primary, which basically makes it brighter, versus the direct immunofluorescence where there can be only like one fluor attached to each of the primary antibodies. But the disadvantage to indirect immunofluorescence is it requires fixed specimens, obviously dead ones. So when we talk about obviously dead specimens, they're not going to be alive and moving. So we can only do a time lapse with direct immunofluorescence and not indirect immunofluorescence because indirect immunofluorescence requires a dead specimen. So you can't see things move around in the cell when you look under a microscope. So yeah, that's a little bit about immunofluorescence. There's also another subcategory of fluorescence microscopy. It's GFP, which stands for green fluorescent protein. And basically, this is just another way to make things glow. I know, there's just so many subcategories. So this green fluorescent protein is a natural fluorescent protein from jellyfish, actually. So the gene from the jellyfish for that naturally fluorescing protein has been isolated and then cloned. And then the gene for GFP can be added to the ends of proteins of interest to tack on a fluorescent tag in live organisms. So just think of any protein and then just attach at the end GFP. And I'll basically make a fusion protein, and the GFP attached directly to the protein of interest will create that glow. It's usually green because just of how the fluor is. It can come in other neon colors like yellow or pink or whatever, but green is the most common. Now the advantages of the GFP is that it's has real-time localization, and there are many types of GFP variants, so obviously you can get rainbows of colors, so you can have like a different color scheme. So let's say you want to make like a legend, like pink means this, green means that, yellow means that. Like you can do that with GFP because there's different colors. Okay, now we're going to talk about another subcategory of fluorescence microscopy. There's so many of them, oh my gosh. This is the last fluorescence microscopy subcategory, so don't worry. This one is called confocal microscopy. And basically the essence of this one is whenever you see a very black background, you'll know it's confocal microscopy because it's all black with only seeing usually 1 to 2 colors. So if we have this image here, you see that the background is black and then the actual image that it's focusing on will be in color. So basically black background equals confocal. So traditional fluorescence microscopy can be blurred from signals coming from above and below the planes of focus, and this makes it really hard to see signals in thick specimens and thick samples when it's not cut like very, very thin. So this type of microscopy uses a pinhole laser beam and basically excites only a small plane of the specimen with fluorescent light. So just think instead of like the whole thing being lit up, it's only that one little laser beam. Wherever that laser beam goes through, it's able to be very precise with the excitation beam into the specimen on that slide. In this way, it is able to produce really crisp single plane images, and it can be used in live specimens as well. We can just see different parts of it illuminating. And honestly, this is a very, very high resolution, and confocal microscopy is really good. The only disadvantage is the price. It's very pricey for this, but it does give a very beautiful high resolution image because it's a single pinhole beam laser. It's very precise and it's able to just pinhole that one thing in that even a live specimen, which is very convenient. I would like to see more of this type of microscopy because it's very high resolution, but obviously because of that it makes it more expensive because of the resources that it needs, that pinhole laser, you know, and it obviously costs a lot for that. But yeah, if you have all the money at your disposal and you want to buy a microscope, buy the confocal one. I don't know, I think it's one of the cooler ones. Anyways, we're moving on to another category. This is not attached to fluorescence anymore. This one is EM, so electron microscopy. I actually saw this one a little bit in biochemistry, but not too much showing up, so now we get a little bit of a deeper dive, which is kind of cool. So electron microscopy also is a much higher resolution because obviously it's a beam of electrons, not like a wavelength of light. It does require dead cells, so not moving, basically fixed cells if you want to think of it like that. There are two types of electron microscopy, so think of these as like subcategories. We have SEM and TEM. So SEM is scanning electron microscopy and TEM is transition electron microscopy. So just a little bit before we go into those subcategories, electron microscopy is 40,000 times better than light microscopy because of the resolution is so high because of these electron beams. So let's first talk about TEM, which is transmission electron microscopy. So the image depends on differential scattering of incident electrons by molecules in the preparation. So basically what that means is if I show you this diagram here, you can see in transition and scanning, both of them are very similar. They have the tungsten, filament, cathode, the anode, condenser lens, beam of electrons. And then here's where it gets a bit different. So in transmission electron microscopy, the electron goes through the specimen, whereas scanning, it does not. The electron bounces off the specimen surface. And SEM also has these scanning coils as well, whereas TEM has a projector lens here. But yeah, transmission electron microscopy goes straight through the specimen, and then it produces this kind of flat single focal plane image. And electron microscopy generally uses heavy metals, but with these flat single focal planes, we can see internal structures as well. So if we compare that to something like SEM scanning electron microscopy, where we don't have a projector lens and this electrons actually bounce off the specimen to the detector, SEM basically can only visualize the surface, whereas the TEM obviously goes straight through the specimen. So it is similar to the TEM, where it looks kind of more flat, single focal plane. SEM looks more 3D, like definitely more 3D, and the surface is coated with a heavy metal to be able to, bounce off of the surface and go to the detector. So basically when you think of SEM, it measures the amounts of electrons scattering off of the surface of a heavy metal coated objects. So you have to coat the object with a heavy metal so it can bounce off, obviously, versus TEM, where it just goes straight through the specimen. And you can generally distinguish between the two. Obviously, it definitely looks more flat with the TEM versus SEM. So that's a big giveaway. Other than that, they're pretty similar in their processes. The only kind of difference is obviously TEM goes straight to the specimen, scanning just bounces off because there's a coat of heavy metal. So yeah, just wrapping up about microscopy. So we talked about light microscopy, which is a subcategory of interference microscopy. So basically interference microscopy, think light microscopy, DIC, which is differential interference contrast microscopy, and phase contrast microscopy. So those three are subcategories of interference microscopy. Then we have the fluorescence microscopy, which we have the subcategories of immunofluorescence, the GFPs, which is the green fluorescent proteins, and then the confocal microscopy, the one that's really expensive that I kind of have a preference for. And then we have the electron microscopy, and two subcategories is TEM and SEM, TEM being the transmission electron microscopy, whereas the SEM being scanning electron microscopy. And just remember, transmission goes straight through and scanning has a heavy metal coated and it bounces off. So the electrons bounce off into the detector. So yeah, this little tidbit about microscopies. I hope you guys enjoyed this video. Not really biochem, but more bio definitely. But if you're going into any kind of scientific field, it's really useful to know the types of microscopies you can use and what's out there. And just compare the price ranges and the resolutions. See what the best option for whatever lab or whatever test you're doing, or if you're in research. So yeah, I hope you guys enjoyed.